Okay, hello everyone, good afternoon. I can see Sakshi and Sunny already live here on the channel, and I hope others are going to join soon. And as you can see, we will be talking about the last part of this chapter, that is a common pathway for carbohydrate, protein and fat metabolism. And then I'm going to take some doubts if you have, and then we will have few questions for the quiz. So we will have a short quiz today and a long quiz tomorrow. So let us wait for everyone to join in. Till that time, Sakshi and Sunny, just let me know that whether this audio is clear or not. Hiren, good afternoon. Is the audio clear? Very good, very good. Because in the morning I had couple of sessions, but that time the students were facing some net issues. Okay, Sunny also says it's all good. Akshay, good afternoon. Is it Akshay or Akshaya? I think it is Akshaya, right? So is this the first class for you? Reshma, good afternoon. Akshaya, okay, okay. So how come it is like so late that you got to know about this channel? And before we start with the chapter, I have couple of things which I want to share with you. That is about the schedule, the next chapter and all those things which we will be taking up. So I'm going to be sharing the schedule with you. Okay, so those who have joined in little late, this is like our almost last chapter, last lecture on this chapter. And we will be talking about the common pathway for carbohydrate, protein and fat metabolism. And then I'm going to take doubts in case if you have. And then I have also planned a small quiz for you. It's like 5 questions, but this will give us some idea. And then tomorrow we have the long quiz, like maybe 15-20 questions, and we will discuss all those questions also. And for those who have joined in late, I was just telling you that I'm going to be sharing the schedule with you for the topics which I'm going to be taking in next 2 weeks time, so that you know you can plan your schedule accordingly. Okay, so let's go over the basic things which we have to talk about, and then I'll come to those schedules and everything. See, this is about the platform that we are using, the channel we are on, that's Let's Crack NEET UG in English. And those who are new students, for them, this is about me. I'm Dr. Neela Bakore, your biology teacher. And we are using India's largest learning platform, that is Unacademy. And I guess now those who have not installed this app yet, they should do it. And the reason for that is, moving forward, I'm going to be taking classes on 2 platforms. One is Unacademy Learning App, where there are special classes conducted, and the second is going to be our YouTube channel, Let's Crack NEET UG English. So there are important chapters which I'm going to start on the app in the special classes also. So if in case if you have not installed this app, you need to do that. Plus subscription, I have already talked about it, that there are benefits of this plus subscription, that is daily live classes, structured courses, quizzes, and unlimited thing, and a recently introduced Iconic subscription. Now this Iconic has additional features. Apart from what is there in Plus, there will be a personalized coach or a tutor who is going to help you. Then your complete curriculum will be planned. There will be test analysis. That means whatever questions, whatever mock test you take, they will be analyzed. You will be given information about what went wrong and everything. And there are benefits of this Plus anyways. So this comparison I have already shared with you. Now this is the subscription plan, and this one is for Plus. So for Plus, the option that you have is 1, 3, 6, 12 and 24 months. And the amount which you are required to pay, you can get 10% discount if you take that subscription by using this code. This is for Plus. And this is for Iconic, and as Iconic has some additional features, you can see there is a change in the subscription fees, and it is for 6, 12 and 24 months. And but here also you can get that 10% discount if you use this code. This is my code, okay, and these are the benefits of Plus. Now let me tell you something about this schedule which I was talking about. Hello Kalai. Alright, see, this is the schedule for my special class which I'm taking on Unacademy Learning app. And here the timing is 10:30 to 12. That means it is a 90-minute class. Normally, if you're preparing for NEET, we have to complete more and more syllabus, so we have to have a little longer duration of the class. And this schedule which I have given you, this is from the 2nd of November, but tomorrow, that is 31st, I'm starting a new chapter on this special. Okay, so if you want, you can join in from then, from tomorrow itself, and if you want, you can start from 2nd, but I would suggest start from tomorrow, because I'm going to be starting with the basic structure of DNA, nucleotides, nucleosides, then purine, pyrimidine, nitrogen bases, and how the bonds and everything is formed. Okay, so this is just for your information so that you don't miss out on any thing. And this is for this particular channel. So tomorrow we have a quiz on respiration chapter complete, and from 2nd we are starting with one chapter of human physiology, and that is breathing and exchange of gases. And the timing is going to remain from 12:30 to 2. Here also I have increased the duration of the class. It's going to be 90 minutes. And see, now let's crack NEET UG English. On this channel, our classes are going to be 12:30 to 2. And this is till 11th, 12th November. After that, I will add things and then I'll let you know the further timetable and the further chapters which we will be starting with. Okay, so note it down what is coming up on the special class and what is coming up on Let's Crack NEET UG English. Alright, now let us come to our topic. Common pathway for carbohydrate, protein and fat. See, we know that carbohydrates, when are broken down, we can write that only for carbohydrates. See, so if we talk about carbohydrates, Especially the carbohydrate that we take is glucose. So what happens to glucose? Glucose is broken down into 3-PGAL. We are writing some intermediates, right? We are not writing everything here because we have to see just the part where proteins can also join in and fats can also join in. So 3-PGAL is formed, 3-phosphoglyceraldehyde. Where is this formed? As a result of first part of glycolysis. Glucose, glucose 6-phosphate, fructose 6-phosphate, fructose 1,6-biphosphate, then it dissociates and then you have these two molecules, dihydroxyacetone phosphate and 3-PGAL. So we have written that 3-PGAL. Then we will write pyruvic acid. We know what happens to PGAL, and then for pyruvic acid changes into acetyl coenzyme A, and this acetyl coenzyme A then participates in Krebs cycle, right? This part we have already studied. This part we are very well aware of. So what happens in carbohydrate metabolism is this. Complete part we have studied. Now suppose instead of carbohydrates, say proteins are being broken down. Then again we will get energy. But how will we get energy and how much energy will we get? So for that, let us say here is this protein. Now these proteins we know are broken down into amino acids. Okay, so amino acids are formed by the breakdown of these protein molecules. Now these amino acids will have to join somewhere in this main pathway. And depending upon where it joins, we will be able to calculate the number of ATPs that we obtain. So these amino acids, they join at 3 places. It can join at pyruvate. Some amino acids join at acetyl coenzyme A, and some amino acids join in Krebs cycle, right? So now, how are the questions asked on this common pathway? I'll give you a simple question first, and then we will take a little complicated one. Are you ready for the question? So I'm writing the question here. First, a protein which has, say, 10 amino acids, out of which 3 join at pyruvic acid and 7 join at acetyl coenzyme A. Then, how many ATPs will we get? Is the question clear? Is the question clear? Let me take you to the previous slide. Here, what we wrote was that in case of normal carbohydrate metabolism, what happens is glucose is first broken down. PGAL is formed, then pyruvic acid is formed, which changes into acetyl coenzyme A, which then participates in Krebs cycle, right? If instead of carbohydrates, a protein is broken down. If protein is broken down, we are going to get only amino acids. Now, there are some amino acids which can join at pyruvic acid. Some amino acids can join at acetyl coenzyme A, and some amino acids can join at in the Krebs cycle. Now where in the Krebs cycle, that we are not sure. It can be fumarate, it can be malate, anywhere. So I have given you a question. The question is, there is a protein which has 10 amino acids. 3 amino acids are joining here and 7 are joining here, right? Can we calculate the ATPs which we get from this protein molecule? If one amino acid joins at pyruvic acid, what will happen? Let us go to the next one. One amino acid joins at pyruvic acid. So pyruvic acid changes into acetyl coenzyme A, which takes part in Krebs cycle, right? Do we know what happens here? We will get 3 ATPs. Are you able to remember? It is the link reaction. And in Krebs cycle, one Krebs cycle is going to give us 12 ATP, right? So one amino acid which joins pyruvate or which changes into pyruvate is going to give us 15 ATPs. One amino acid. And how many amino acids are joining here? So 15 into 3. Okay, these many amino acids. Now let us talk about this one. If one amino acid changes into acetyl coenzyme A and this participates in Krebs cycle, so what we get is only 12 ATPs. So how many? 12 ATPs, and how many amino acids are changing into acetyl coenzyme A? Is 7. So are you able to calculate the number of ATPs? Can you calculate the number of ATPs if a protein is joining the common pathway? That means glucose to PGAL to pyruvic acid to acetyl to Krebs cycle. It's a common pathway. Carbohydrates also do this or go through this pathway, and protein, if broken down, will also go through the same pathway. Is it clear? Is this clear? For protein. If yes, then we will take up fat. If fat is broken down, then what happens? Yes, okay, so Hiren says it is clear. What about others? Kalai also understood this. 7 in 3. See, these 3 will undergo link reaction because pyruvate, they are changing into pyruvic acid. So if pyruvic has to change into acetyl, then link reaction will take place. But these 7 are directly changing into acetyl coenzyme A. So link reaction will not take place. Clear? Okay, so now if fat molecule is broken down, what is the fat made up of? Fat when breaks, it breaks into glycerol and fatty acid. These are the 2 things which are formed. Okay, Hiren, Kunal, Reshma, everybody is saying that it is clear. Now this glycerol, it gets converted into 3-PGAL. One glycerol molecule will get converted into 3-PGAL, and fatty acids will get converted into acetyl coenzyme A. Now listen to this question carefully. This will also help me understand whether you have done biomolecules or not? I guess many of you have done this chapter of biomolecules, so I will also be able to understand whether you have done it properly or not. How many ATPs will be obtained if a diglyceride fat enters the common pathway? Okay, is the question clear? Harish, I guess you've joined little late. So before we end the class, I'm going to again share the schedule for the next 2 weeks with you, and you will know what is the timing and which chapter we are planning to do. Okay, answer the second question now. I'm taking you back to this original slide, and here the question is that 1 diglyceride fat, if it enters the pathway, then how many ATPs will you get? Can you tell me how many ATPs will you get if a diglyceride fat breaks down through this pathway? Now let me ask you one question. Do you know what is a diglyceride fat? Who knows about diglyceride fat? This is in biomolecule chapter. In 11th class, the unit where we study cell cycle, cell division, herein says 2 fatty acid chains attached to what? Herein, these 2 fatty acid chains are attached to what? And suppose to glycerol. Very good. So if this is glycerol, do you know the formula of glycerol? So I'm going to write this diglyceride fat for you here. CH2OH, CHOH, CH2OH. This is glycerol. Now to this glycerol, because there are 3 OH, 1, 2 or 3 fatty acids can attach. So if this is a fatty acid, a long chain hydrocarbon, it is acid, so it has to have COOH, right? Now this will be lost as water molecule, and the bond which is formed is called ester bond. So if to glycerol 1 fatty acid is attached, if to a glycerol 2 fatty acids are attached, or if to a glycerol 3 fatty acids are attached, this will be called monoglyceride fat. This is called diglyceride fat, and this one is triglyceride fat. Okay? So now the question was that if a diglyceride fat is broken down through this pathway, then how many ATPs will we get? So if this fat is broken down, you will get 1 glycerol and you will get 2 molecules of fatty acids, because this is a diglyceride fat. Now where is glycerol joining? Glycerol is joining at PGAL. Herein is saying 44 ATP. From 1 diglyceride fat we will calculate. See, from PGAL to pyruvic acid, here what do we get? We get 2 ATPs, and and we also get one NADH2. So that means here we are going to get 5 ATPs, okay? Pyruvic acid to acetyl coenzyme is a link reaction, so we will be getting 3 ATPs because we get NADH2 here, and this is Krebs cycle, which is going to give us 12 ATP. So 12 plus 3, 13, 14, 15 plus 5. So if one glycerol is broken down through this pathway, we will get 20 ATPs. Rahul, good afternoon. Is this answer 20 clear to everyone? How did we get 20 ATPs if one glycerol joins this common pathway? Okay, Hiren also says that it is 20 ATP, that means you have calculated. Now, fatty acid directly changes into acetyl coenzyme A. That means it will only undergo Krebs cycle, and we know Krebs cycle gives us 12. So there are 2 fatty acids, so it will give us 12 into 2, 24, and then you add these two. So that is how Hiren has given 44. Is it clear to everyone how we calculate ATP gain through a common pathway for proteins, fats, carbohydrate? We have done anyways in detail. One glycerol, 20. Now quickly tell me, if it was a triglyceride fat, then how many ATPs will you get? If it is a triglyceride fat, give me the answer. If it was a triglyceride fat, Hiren says 56. In triglyceride, there is one glycerol and there are 3 fatty acids. Glycerol gives you 20, fatty acids are 3, so it is 12 into 3. Total? Very good. So Kalai, Hiren, all of you got the answer. So now you understand how there is a common pathway. Mainly it is carbohydrate which is broken down through this pathway, and all the stages we have studied in detail. But if protein and fat, they also enter the same pathway, then we get different types of ATP numbers depending upon which type of amino acid, which type of protein or which type of fat is getting broken down. Simple? So the carbohydrate pathway that we have talked of, that is glycolysis, link reaction, Krebs cycle, is a common pathway. And in this pathway there is breakdown also and there is building up also, and that is why you call this pathway complete. Respiratory pathway, cellular respiratory pathway, as amphibolic. Anyone who has any doubt in this part so far? This is the last thing that we have to talk of, and I will tell you one more extra thing. And that extra thing is, there is an alternative to glycolysis. And it is known as PPP or HMP. It is called pentose phosphate pathway, or it is also known as hexose monophosphate pathway or shunt. These are the alternatives for glycolysis. So normally glycolysis will take place, but in case there is no glycolysis, or if there is some kind of a problem, then there is an alternative pathway also available. So HMP or PPP, pentose phosphate pathway, or hexose monophosphate pathway. So this completes our chapter for respiration in plants, or we can call it cellular respiration. Okay, so now what we will do is, I'm going to wait for your doubts. And you know, in competitive exam, this type of questions can be asked, not very commonly asked, but yes, they can be asked, and they ask where the fat gets added, or where does it join the common pathway. So anyone who has any doubt in this common pathway thing and how to solve these questions? Go over this one more time. Amino acids can join at 3 places: pyruvic acid, acetyl coenzyme A, or directly in Krebs cycle. Okay, Hiren says no doubt. And fat is going to, because it breaks down into glycerol and fatty acid, it can join at 2 places, either 3-PGAL or acetyl coenzyme A. So depending upon how many fatty acids are there, you can count the number of ATPs which will be produced. So Hiren, you don't have any doubt. What about others? Is this part absolutely clear? See, this chapter is important. If you are in class 11th, then either glycolysis or Krebs cycle, or even electron transport chain, or even oxidative phosphorylation, any of these 4 can be asked as a 5-mark question. So this, if you have understood everything clearly, you can easily get 5 on 5. And in the competitive exam, that is NEET, the questions are asked like from electron transport chain, like where is a succinate hydrogenase located, or what is in complex 1. So for that also we need to understand the whole process. Okay, so Hiren, Priya and Kalai, everyone has understood everything. Okay, now I'm just waiting for your doubts before I give you few questions. Doubt in the entire chapter, starting from types of respiration, aerobic, anaerobic, everything. So if there is any doubt, you are free to ask those questions so that we can discuss this. And if there is no problem, no doubt, then we can start with the quiz. Actually, I have just framed 5 questions for you. And after every question, we are going to discuss that question in case if you go wrong. But before that, I want to know if there is any confusion of any kind in this chapter. Hiren is asking types of fermentation. Okay. See, fermentation term is given to anaerobic respiration or breakdown. Now there are 2 possibilities. One, you have homofermentation or heterofermentation. Homofermentation is only one product, and hetero means more than one product. And if we are talking about homofermentation, then lactic acid fermentation is the answer for this. And if we are talking of hetero, then alcohol fermentation is the answer for this. Just types, okay? So Hiren, you got the answer, just the types, homo, hetero. You can call it lactic acid fermentation or alcohol fermentation. Clear? If it is clear, then I'll take the question from Reshma. Okay, now Reshma's question is calculation of ATP in shuttle system. There are two shuttle systems that we have talked of. One was glycerol phosphate shuttle system, and the second is malate aspartate shuttle system. What exactly is a shuttle? Shuttle word is used for some vehicle which is, you know, moving things from one place to another. Like if there is a big park or something, then there are shuttles which run short distance travels. So here, suppose this is the membrane of mitochondria, which is impermeable to NADH2. Here also there is a membrane of mitochondria which is also impermeable, and as a result of glycolysis, two NADH2s were formed in the cytoplasm. So this outer part is cytoplasm and this is the mitochondrial membrane, and inner part is obviously matrix of mitochondria. These two NADH2, which we calculated as worth 6 ATPs, they will give us ATPs only if they enter the electron transport chain and oxidative phosphorylation. But they cannot go in. So what happens is they dissociate into NAD and a pair of proton. But here we are talking of 2, so everything will be doubled. When this proton has to be pumped in, energy is required, ATP. So to pump one proton pair in, 1 ATP is required. From one NADH2, 1 pair, 1 ATP. From the second NADH2, 1 more pair of proton, 1 more ATP. So we got 38 ATPs in that energy balance sheet which we made. So -2 to pump the proton pairs from cytoplasm to the mitochondrial matrix. And that is how we get 36. This is part 1. So is this part clear, Reshma? And then we will come to the second part. If it is glycerol phosphate shuttle, then the proton pair which has entered is accepted by FAD and we get FADH2. Okay, so we get FADH2. If the molecule which has given protons is NADH2, which was worth 3 ATPs, and the protons are accepted by FADH2, which is worth 2 ATPs. So 1 NADH2 changing into FADH2, ultimately you get 1 ATP less. So if 2 NADH2 will give you 2 FADH2, 2 ATPs less. So anyways we were getting 36. Now because of this shunt or shuttle system, -2, because you are not getting NADH2, you are getting FADH2s. Clear? And if this is the shuttle system, then when the proton comes here, this proton is accepted by NAD, so you will get NADH2. So outside was also NADH2, inside is also NADH2. So there is no change in the energy or ATP calculation. So we will still get 36 ATPs. Okay? So is it clear now? Kalaya is asking, where do we get FAD and NAD in both glycerol and, see, the NADs which are in the cytoplasm, they remain there. They cannot go inside mitochondria. The NADs, FADs which are present inside mitochondria, they remain inside mitochondria. They cannot go out. So in cytoplasm also there is NAD, FAD, and in matrix also there is NAD, FAD. Only thing is they cannot move across the mitochondrial membrane. Okay, so Reshma says it is clear. Any other doubt? Shuttle system done, types of fermentations done. Okay, Hiren says clear. Kalai says, Kalai, did you get the answer to your question? Are you satisfied with the answer? Where are these NAD, FADs there? Why glycerol is accepted by FAD and not NADH2? Where is glycerol accepted by FAD? This is called the glycerol phosphate shuttle system. The protein which is there is actually glycerol phosphate shuttle. Is it clear? In glycerol shuttle system, see, the shuttle system is a protein which is in the membrane, and that protein is just helping in transfer of the protons. Okay, can you see me? Because here on my phone there is some, I think, problem here. Are you able to see me clearly? Okay, now it is again good. All right, so yes, perfect. So I think you're all good, no doubts, no confusions. So shall we take few questions just to understand whether this chapter is clearly understood by everybody or not? And I have decided only 5 questions for now, and because after every question we have to just discuss it, and that's why. All right, this is the first question. Let me see how many of you can answer this. Can you see this question? Okay, I have started getting the answers also. Reshma says B, Hiren says B, Kalai also says B. Okay, we are talking about RQ, that is respiratory quotient, and we know that for fats and proteins it is always less than 1. Perfect. So everyone has given me the correct answer. Okay. Second question. Can you see the second question now? And write down the number, question number, because otherwise I'll think you are giving me the answer for second, but you are giving me for the first. Question number 2. In anaerobic glycolysis, net gain of ATP is, Reshma says A, Kalai says A, Hiren 2, okay, that is, oh, answer is 2, okay. Krish also says question number 2, answer is A. And this answer is absolutely correct, because only 2 ATPs are obtained, those 2 NADH2s get used up while the second part of the reaction. Okay, the third one, third question. Question number 3. Okay, Reshma has given the answer as B. Let me see what is the answer from others. Hiren says cytoplasm. Okay, that is B. Kalai says answer for 3 is A. Krish says D. Okay, so we have 3 options here. We have B answer, we have D answer, and we also have A answer. Okay, anyone else who is giving me some other answer? Kala is saying third answer is C. Oh wow, we have all 4 options here now. So some of you have given B, some has said D, some has said A. Okay, now let us see where are we going wrong. What is EMP? Can anybody tell me what is EMP? If you know the full form, you automatically will know the answer. Kalai is now saying 3, answer is C, okay? Hiren is saying glycolysis. See, it is Emden, Meyerhoff, Parnas. Emden, Meyerhoff, and Parnas. This is, or these are the names of the scientists who actually gave us the pathway of glycolysis. And where does glycolysis take place? In cytoplasm. So all those who were saying D, A, C, I don't know what was the confusion. Tell me what was the confusion. What were you thinking? If you are talking of inner mitochondrial membrane, what takes place in inner mitochondrial membrane is ETC. What takes place in mitochondrial matrix is Krebs cycle. So what was the confusion? I guess you were not able to remember that EMP, that is Embden-Merov-Parnas pathway, is another name for glycolysis. Yeah, Samuel, hi. So is this question clear now, all those who gave incorrect answer? I guess they were not sure about EMP, or they were not able to recall EMP immediately. All right, I'm giving you next question now. This is the fourth question. And now give me the answer for this. Fourth question. Oh, Krish says, I wasn't knowing that it is also, yes, it was given by three scientists, Embden, Merhoff and Parnas, and that is why it is also known as EMP pathway, okay? Reshma says fourth, answer is B, okay? Kunal is writing something, mitochondria में Krebs link and oxidative phosphorylation. What exactly you want to know, Kunal? Okay, Reshma has given B, Kunal says 2, Hiren says fourth is 2, and Krish also says 2. Again, if now you know, those who are not aware of it, now you know we are talking about glycolysis. And in glycolysis, we get 2 molecules of NADH2 and 2 ATP. So here our answer is 2 NADH2s. Yes, Kunal, you were asking some question. I'm not able to understand. It says Krebs link and oxidative phosphorylation. Okay, you frame your question, and till that time I'm going to give you one more, and this is the one. The fifth question. Okay, answer for fifth. I said, ma'am, in mitochondria, link Krebs and oxidative phosphorylation takes place. In mitochondria link, oh, okay, you were giving the location. All right, got it. Padmavati is saying you did not understand. What was that thing which you were trying to understand here? Hiren has given the answer as A, Krish has given the answer as C, Kalai has also given the answer as C. The fourth question, okay, I'll go over the fourth one, alright? So for Padmavati, once we are done with this, I'll go back to the fourth question. Okay, so I have 2 answers so far for this question, and that is A and C. These are the 2 answers which I have, options rather, which I have received so far. Reshma says A. Okay, so we have that option written here. Now what exactly is TCA cycle? What is TCA cycle? Tricarboxylic acid cycle. Tricarboxylic acid cycle. And what does this mean? That an acid which has 3 COOH groups. So that means we are talking about citric acid. So the first compound formed is citric acid, which is a 6-carbon compound. And how is that formed? Acetyl coenzyme A is accepted by, see, I know some simple formula making options. See, I know some simple formula making options. So, which one is the primary acceptor? It is oxaloacetic acid. Is it clear? So those who thought it was citric acid, you were talking about the product. The primary acceptor means whatever is entering into the cycle, who is accepting it? And what is entering into the cycle is acetyl coenzyme A. And it is accepted by oxaloacetic acid. Citric acid is the, this mnemonics is printed in my head. Very good. So if you know this, you can never go wrong. Alright, let me go back to the fourth question. Padmavati had some doubts. See, the number of NADH2 molecules in EMP pathway or glycolysis, in EMP pathway or glycolysis, we get 2 NADH2 and 2 ATP. Have you studied glycolysis? Did you study glycolysis, Padmavati? If yes, then you would know this, and if not, then I would suggest that go through the video which I made on this glycolysis. We did a complete 1-hour lecture for glycolysis and we went through each and every step. Kunal is asking about the mnemonics. It was see, I know some simple formula making options, and this accepts acetyl coenzyme A. This is oxaloacetic acid, this is citric acid, this is isocitrate, this is alpha ketoglutarate, this is succinyl coenzyme A, this is succinate, this is a fumarate, this is malate and oxaloacetate. So these are the intermediate compounds in Krebs cycle. And because Krebs cycle is little complicated, this helps us understand. And if you have not watched this video also, then please do that, so that where ATP is produced, where NADH2 are produced, where FADs are produced, all those things will be clear, absolutely clear to you. Okay, so now Padmavati, there was a question from your end. So, did you study glycolysis earlier? As I guess this is your first class, you have not attended the previous lectures, so just go through those lectures. You will find them on the channel as recorded lectures, and just go through all those videos which are there of this chapter. So there is one on glycolysis, there is one on Krebs cycle, and these are very, very, very important for not only for your 11th class exam but for NEET also. So if you are an 11th class student, then also you need to know it in detail, and for NEET obviously we need to know everything in detail. Okay, so this was the 5th question which we did, and just simple questions, simple 5 questions I had picked for you, but tomorrow the questions will have little more difficulty level. Okay, so I guess the chapter is absolutely clear, because when I asked for doubts, nobody asked me doubts, so that means you have understood everything clearly. And tomorrow we are going to have the main quiz. That means the questions which are like slightly more difficult. Now don't forget to subscribe on the channel, don't forget to take the subscription, and while you take the subscription, don't forget to use this code so that you get the discount. Now I'm taking you back to the schedule sheet so that the students who joined in little late, they will also be able to see it. See, this is on the channel Let's Crack NEET UG English, the same channel where we are talking right now. Okay, so on this channel we will be, tomorrow is quiz, that is 31st. First is Sunday, so it's a break day for us. 2nd November we are starting with one chapter from human physiology, that is breathing and exchange of gases. And also remember the timing. The timing is 12:30 to 2. The duration is 90 minutes. Otherwise we won't be able to complete the syllabus. And this is the special class which is conducted on the Unacademy learning app. So if you have not installed the app, go to the app store, install this app. These classes are also free, and here I'm starting a chapter, molecular basis of inheritance. Tomorrow we have the first lecture of this, that is 31st. First again we don't have anything, and from 2nd November everything is in front of you. So I've given you the schedule for 2 weeks, and then I'll give you the other chapters also. Yes Kunal, we will take biomolecules also slowly and gradually. And one more very important thing. Tomorrow 31st, our class on the channel Let's Crack NEET UG English is at 12:30. Okay, and from 2 to 3 I have planned a special session for all of you on a channel called Biology 101, and this session is on strategies to score 360 on 360 in biology in NEET. So don't forget to join in both these sessions. And tomorrow we will have the quiz. Timing is 12:30. What was your doubt, Kunal? You wrote that I said, ma'am, in mitochondria link, Krebs and oxidative phosphorylation takes place, which is correct. So in my outer mitochondrial space, link reaction takes place, Krebs cycle takes place in the matrix, and oxidative phosphorylation takes place in oxysomes. Okay, so again, still collect your doubts. Tomorrow before we start the quiz, again I'll give you 5 minutes to ask doubts. Alright, so I'll see you tomorrow at 12:30 and then again at 2. So till that time, revise the chapter, come up with the doubts and ask those doubts before the quiz. So bye for now, take care.